sada nam je na redu naš profesor Adnan Čustović. Ja ću za početak pročitati njegovu biografiju na našem jeziku, pa na engleskom, tako da zamolit ću profesora da se pripremi. Ja ću prestati da šerujem svoj ekran, tako da može profesor da šeruje svoju prezentaciju. I profesor, izuzetno je zadovoljstvo što ste danas tu sa nama. Hvala vam puno. Najcitiraniji i najpriznatiji bosansko-hercegovački istraživač je pedijatrijski alergolog profesor Adnan Čustović sa Imperial College Londona, koji ima čak 52.000 citata i H-indeks od 106. Diplomirao je na Medicinskom fakultetu Univerziteta u Sarajevu 1985. godine kao jedan od najboljih u generaciji. Na istoj ustanovi magistrirao je eksperimentalnu medicinu. Njegovo stručno usavršavanje se sastojalo od specijalističkog usavršavanja iz pedijatrije od 87. do 91. i uzastopnih imenovanja kao saradnik kliničkog istraživanja i specijalista za alergije od 92. do 98. godine. U tom periodu mu je dodijeljena nagrada MR 91. godine, doktor medicine sa zlatnom medaljom 96. godine i DR 2000. godine na Univerzitetu Manchester. Nakon što je završio svoju kliničku specijalističku obuku, dobio je stipendiju za istraživanje za Asthma UK. Unaprijeđen je u poziciju profesora 2002. godine. Bio je profesor alergije na Univerzitetu u Manchesteru do septembra 2015. godine, gdje je radio kao vodeći Centra za respiratornu medicinu i Institutu za translacionu medicinu. Sarajlija Adnan Čustović je naučnik svjetskog glasa koji je dobio niz priznanja, među ostalim zlatnu medalju Evropskog respiratornog društva za istraživanje astme te BSACI William Franklin medalju za izvanredan doprinos kliničkoj alergiji i nagrada predsjednika CPP za istaknuta dostignuća u liječenju dječje astme. 2020. godine izabran je kao fellow Britanske akademije medicinskih nauka. Rezultat njegovih istraživanja o astmi i alergiji primjenjuje se u praksi svakodnevno. On predvodi grupu za pedijatrijsku alergiju u okviru Centra za pedijatriju i zdravlje djece na Imperial College Londonu. I počasni je konsultant za pedijatrijsku alergiju u bolnici St. Mary i Royal Brompton bolnici. Njegovo istraživanje fokusiralo se na porijeklo i prirodnu istoriju astme i alergija kroz životni tok s naglaskom na prevenciju i prevođenju za dobrobit pacijenata. Njegovi nalazi i istraživanja su od velikog praktičnog značaja, te su informirali i promijenili nacionalne i međunarodne smjernice o prevenciji i liječenju astme. Njegove studije o alergijama na hranu značajno su uticale na kliničku praksu. On je bio pionir u korištenju metodologija vođenih podacima u analizi složenih podataka, uključujući prvu upotrebu mašinskog učenja u respiratornoj medicini, to jest alergiji. Nosilac je počasnih profesorskih titula u Manchesteru, također u Cape Townu, Singapuru i Zagrebu. The most cited and recognized Bosnian researcher is the pediatric allergist Professor Adnan Čustović from Imperial College London, with 52,000 citations and age index of 106. He graduated from the Faculty of Medicine, University of Sarajevo in 1985 as one of the best of his generation. At the same institution, he obtained a master's degree in experimental medicine. His professional training consisted of residency training in pediatrics from 87 till 91 and successive appointments as a clinical research associate and allergy specialist from 92 till 98. During that period, he was awarded Master of Science 91, Doctor of Medicine with a gold medal 96, and Doctor in 2000 at the University of Manchester. After completing his clinical specialist training, he was awarded an Asthma UK Research Fellowship. He was promoted to professor in 2002. He was professor of allergy at the University of Manchester until September 2015, where he worked as center lead for respiratory medicine at the Institute for Translational Medicine. Adnan Čustović is a world-renowned scientist who received a number of awards, including the European Respiratory Society's Gold Medal for Asthma Research, the BSACI William Franklin Medal for Outstanding Contribution to Clinical Allergy, and the CIPP President's Award for Outstanding Achievements in the Treatment of Childhood Asthma. In 2020, he was elected as a Fellow of the British Academy of Medical Sciences. The results of his research on asthma and allergy are applied in practice every day. He leads the pediatric allergy group within the Center for Pediatrics and Child Health at the Imperial College London and is honorary consultant in pediatric allergy at St. Mary and the Royal Brompton Hospital. His research has focused on the origins and natural history of asthma and allergies across the life course, with an emphasis on prevention and translation for patients' benefits. His research findings are of great practical importance and have informed and changed national and international guidelines on asthma prevention and treatment. His studies on food allergies have had a significant impact on clinical practice. He pioneered the use of data-driven methodologies in the analysis of complex data, including the first use of machine learning in respiratory medicine and allergy. He holds honorary professorship in Manchester as well as in Cape Town, Singapore, and Zagreb. Professor Čustović, it is our pleasure to have you with us today. Please, if anyone has any question, you can write it in a chat. And Professor, I'm going to stop sharing my screen for now so that you can start sharing your screen.
Ok, nadam se da vidite moj screen i da me čujete dobro.
Čujemo vas dobro, screen još uvijek ne vidimo.
Evo, sad ste trebali da vidite.
Tu je, sad će. Tu je, super, sve vidimo.
Odlično. Prije svega hvala vam na pozivu, mislim, za sudionike, organizatori su zamolili da ovo uradim na engleskom, tako da će predavanje biti na engleskom, vjerovatno malo jednostavnije za mene, ali nema veze, sljedeći put će biti na našem. Prije svega, da se zahvalim Ediju. Edi, BH, Future Foundations, apsolutno nevjerojatna inicijativa. Mislim, ako nas išta može spasiti, to je znanje i znanost. Ja se lično mislim da imamo isto prezime, ja se lično zbilja ponosim time da je jedan Čustović to uradio. Hvala ti za sve što si uradio, za sve što radiš i za sve što ćeš uraditi. Zbilja briljantno. Hvala i svim sudijama i naravno sponzorima bez kojih to ne bi bilo moguće, no zvijezde dana ste naravno svi vi. Hvala vam što ste imali hrabrosti, što ste imali strpljenje i što želite nešto da promijenite. I gledajući sve to, ja nemam nikakve dileme da je budućnost Bosne i Hercegovine puno svjetlija nego što mnogi misle i vjerovatno puno svjetlija nego što mnogi bi to i željeli. Ja sam zbilja duboko dirnut ovim pozivom i hvala vam svima. I nadam se da ćemo se vidjeti uživo u Sarajevu 3D ili bilo gdje drugo u skorijoj budućnosti. And now to move it to kind of English and to the topic of my presentation. I don't know whether you have screens and whether you can see me on your screen. If not, we'll do something about that as well. But I've kind of deliberately decided to talk about mathematics and machine learning and AI because there will be plenty of you who are not really in medical research. But the whole idea of kind of splitting science into different silos just doesn't make any sense, because what we're trying to do is to advance human knowledge for the benefit of all of us, for the benefit of humanity. Now, these are my potential conflicts of interest. I don't really think they're any relevant to anything that I'm going to be talking about today, but you know, you're all intelligent individuals, you'll be able to make your own opinion about that. My only kind of real conflict of interest is that despite I'm a medic, so I'm a practicing doctor, pediatrician, I kind of really love mathematics. And I'm going to talk to you today about asthma. So why asthma? Well, it's the most common complex disease in childhood and now in adulthood globally. I mean, vast amounts of children and adults, up to 15% currently have asthma diagnosis. Now, interestingly, despite being that common, the way we currently diagnose disease is based on predominantly clinical criteria, which really haven't changed for about 2 millennia. Now, we consider asthma to be a single disease, but in reality, it is a set of symptoms that may be caused by a range of different underlying mechanisms. Now, why would that be important? Well, think about fever. Let's say you have a kid with fever 38.7. That's kind of phenotype. That's what we see. The question here is, what is the disease? What is causing this? Because obviously, if it is virus fever, the treatment will be very different compared to bacterial, where you will give antibiotics, or if you sit in, clinic in Africa, it will be malaria. So the same question can be asked for asthma. If this is an umbrella diagnosis for a syndrome comprising a number of different underlying diseases, then our current approach of pushing patients with these multiple disease subtype under a single umbrella and forcing them into a single treatment is fundamentally flawed. So while, like in most areas of medicine, we kind of say, well, we want to deliver a personalized medicine, what the treatment that really that child deserves, or at least stratified medicine, what we're delivering is blunderbuss medicine where one size fits all. And I want to put to you that the only way to move away from this one size fits all approach is to understand the mechanisms underpinning asthma heterogeneity. So in individual patients, which are the mechanisms leading to symptoms? Because if we crack that, if we understand mechanism, then it is fairly easy to develop biomarker of that mechanism and mechanism-based treatment. So the key to unlocking the era of precision medicine is to understand the mechanism underpinning asthma heterogeneity. Now, this sort of conceptual framework is applicable to most other complex diseases, from a number of different cancers to arthritis to diabetes. So I'm using asthma here as an exemplar. And what I would like to ask today is a question whether machine learning and AI, which have been in press so much recently, can help us understand the progression of asthma and underpinning mechanisms. Now, why machine learning? Well, you will all be aware, and you have seen it, and you have experienced it firsthand, that in the last 10, 15, 20 years, our ability to generate data has increased exponentially. So now we can collect vast amounts of longitudinal data, high throughput technologies, and this ultimately should help us understand the complexity of developmental pathways of common complex diseases. The problem, of course, with having vast amounts of data, that it's challenging to process, even trickier to analyze, and sometimes almost impossible to interpret. At the moment, when faced with data dumps and deluge of data, you kind of scratch your head and think, well, what is precisely that we're looking for? What are the questions that we should be asking? And I think within this context, machine learning offers us a fundamental technology that may help us meaningfully process these large amounts of data that exceed our capacity to comprehend, to analyze them, and ultimately translate that into increased knowledge and understanding of mechanisms. So in the context of what I'm going to be talking about today, machine learning models really are used to digest large amounts of data, relatively quickly, identified patterns within this underlying data set, with the idea that these underlying latent patterns are a reflection of different disease mechanisms. One has to say right at the start, the clusters that we identify using these modern technologies are informative, but will not necessarily directly correspond to underlying biological pathways. So a lot of people today say, oh, we've described these clusters and they are underlying disease endotypes. Well, there is a step from understanding the patterns within the data sets to understanding mechanisms and translating that to benefits of a patient. So the first question is, can you trust scientific discoveries made using machine learning? Well, as a first step, one should at least try to verify it. The other way that I look at the potential of machine learning in bioscience very much aligns with a collaborator of ours, John Wynn, from machine learning group at Microsoft Research in Cambridge. Most use of machine learning and AI in medicine today is about predictive machine learning, trying to predict disease or predict outcome. And in this context, you're trying to make prediction for individual patient, but without really requirement to explain this prediction or underpinning mechanisms that explain the process. And it's very common with building all these automated systems where explanations are not needed. Question, which type of disease does this patient have? How it's going to progress? What does this scan mean? Personally, I'm much more interested in another area, which is explanatory machine learning, where the goal is not just to describe these underlying patterns, but also to understand them, to understand the processes that gives rise to the patterns within these data sets. And I'm going to give you three stories, three exemplars, going from our early work that took us beyond sort of allergy or allergic sensitization, towards understanding how different patterns of allergic sensitization make individual children susceptible to completely different diseases. Then I'm going to tell you a story of these phenotypes and finally end up with sort of bread and butter of allergic diseases, which is a story of atopic march. And all of these are ongoing stories, ongoing research ideas. And I hope that at least some of you will find bits of that interesting and perhaps take part in the ongoing and future research. Most of the data that I'm going to show you today has been generated within the UK Stellar or Unicorn Birth Cohorts Consortium, which I lead with a number of very close colleagues and friends, which comprises 5 birth cohort studies. What's birth cohort? It's a study where we follow individuals from before birth, sort of 8th week of pregnancy. Now we have data up to adulthood, early 30s. So a vast amount of longitudinal clinical data from 5 birth cohorts: Manchester, Aberdeen, Kent, Bristol, and Isle of Wight. So you have data from about 16,000 individuals followed over a period of almost 3.5 decades. All of the data then being harmonized and integrated in knowledge management platform, which we developed called Asthma eLab, which contains all of the data from all of the subjects from which we can get data extracts. But also within this platform, we can do collaborative and joint research where investigators actually talk all of the time and share ideas, activities, results, codes, you name it. So it's really collaborative modeling and gives you an opportunity to almost do real-time research and collaborate with your peers. And the first story that I'm going to tell is that of taking us beyond allergic sensitization. It's a story of modeling childhood allergy to see how this relates to development of asthma and other diseases. So just a little preamble. The way we currently diagnose allergic sensitization is by either measuring allergy antibodies in blood or doing skin prick tests. Yeah, and then based on the results, we say, well, this child is sensitized, and then very often it is translated into this child is allergic to allergen A, B, or C, based on, criteria which are almost universally completely arbitrary, and sometimes which don't make sense. For example, the cutoff point in blood tests that we use that differentiates between sensitized, i.e. allergic, and non-sensitized, i.e. non-allergic child is actually the lower detection limit of the assay as it was developed almost 40 years ago. So clinically, it doesn't make sense. And, sitting in the clinic, you see these kids who have a single positive test with tiny little wheel, or you see kids with a number of different positive tests, really large skin test reactions. The timing of development of this allergy antibody response differs between different children. And we ask question, well, does the pattern of allergens that you're allergic to and the timing where the IgE, allergy antibody response arises, actually tell us something fundamentally important about this feature that we call allergic sensitization? And we were very lucky at the time, so this is probably about 20 years ago, by a pure chance, we got in touch with colleagues in computer sciences and machine learning in Microsoft Research, Cambridge, particularly a gentleman called Chris Bishop, who is one of the grandfathers of machine learning and author of critically important, probably the best textbook on machine learning. And we were discussing these ideas of how to move from standard tools towards greater understanding of patterns in the data and translating this into understanding of mechanisms. So together with them, we then moved in the direction of Bayesian inference and graphical modeling of a problem space to try to understand what are the underlying patterns. So in our birth cohorts, we have data on skin tests and on blood tests longitudinally to a number of different allergens. So we ask the question, does the pattern of allergens that you're sensitized to, the size of the response, the timing of response, actually tell us something about allergic sensitization? This is early days of Bayesian inference, very early days of graphical modeling, but together with experts around the world and colleagues in Microsoft Research, we created a graphical model of problem space, not only based on the data that we had, but also on the overall prior knowledge that we had. The probability of when sensitization is likely to occur, how it's going to develop. This is not black box model. This is not throwing the data, something is going to come out. This is a graphical model that captures prior knowledge. Now, of course, the methodologies at the time were not there, but in Microsoft Research, they developed infer.net, which is a framework for running Bayesian inference and graphical models. Of course, it wasn't really designed for medical research, but for financial sector to predict how the shares are going to go up and down. But we were very lucky to have engineers like John Guyver who adapted this framework so that we can utilize it to ask important questions about medicine. And after a couple of years of work and several extraordinary fellows coming from MIT, including Vincent Tan, we prepared the model, derived the code, and ran the model. And the outcome was quite extraordinary, because what the results told us was that there was no such thing as allergic sensitization. Children whom we considered to be sensitized clustered in 4 fundamentally different classes that were characterized by differences in size, in timing on onset, and patterns of responses to individual allergens. Now, initially we gave them names based on, what we think was going on, which was probably wrong. Let's call them allergic sensitization 1, 2, 3, and 4. Now that's interesting, but what was really interesting was that these different clusters of sensitization had fundamentally different association with the disease, in that only one of these clusters of sensitization, which comprised not more than 1/4 of sensitized children, was associated with asthma. But the strength of that association was extraordinary. You know, if you have this pattern of sensitization, you're 22 times more likely to have asthma. So this is not a ratio of 1.2, 22 times more likely. And not only that you're more likely to have asthma, but if you have asthma, you're much more likely to end up in a hospital with acute asthma attack, both you know, by age 3, by age 5, much more likely to have acute asthma attacks, much more likely to have poor lung function, reactive airways, and inflamed airways. So not all sensitized children are at risk of having asthma and having severe asthma, but only some of them. So there clearly are pathologic, really important subtypes of sensitization, and those that are benign or really not related to disease. So at the moment, I can determine for individual patients which type of sensitization they're in, only in a context of study where I have 10, 15-year follow-up and vast amounts of individual to do modeling. What we're currently working on is, how can I do this with a simple test that can be administered at the bedside on a single consultation. So hopefully these new studies and new methodologies, new algorithms for dealing with complex atopy data will allow us to really translate that into clinical benefit. So this took us beyond atopy into a new realm of different subtypes of allergic sensitization and differentiations between important one and less important one for individual patients. I mean, the other story that I would really love to tell you, and I started by saying, well, there are real differences under the umbrella diagnosis of asthma. There probably are different diseases, is trying to understand the different wheezing phenotypes in childhood. And wheezing is cardinal, most important symptom of asthma. And with this, I'm going to take you back 35 years to the article that really kind of was fundamental for persuading me to go down the route of pediatric respiratory medicine and allergy. It's an article written by Nicola Wilson, who was at the time working at Hammersmith Hospital. This is the day and age where all of the drug companies, pharmaceutical industry, was trying to push forward the concept that all individuals who present with wheezing should be considered to have asthma and should be prescribed inhaled corticosteroids. And Nicola Wilson stood back and wrote this rostrum article. And I read it 35 years ago, and I thought it was extraordinary. With the benefit of hindsight, I consider it even more extraordinary now. Everything that she said then stood the test of time and stands today. She's saying the description of wheezing illness in preschoolers has always been confused. Well, nothing has really changed. The association between early life infections and asthma remains controversial, then and now. She then argued that within different individuals, there are different types of predisposition or responsiveness. Allergic one, viral one, bronchial hyperresponsiveness, and that different combinations of those give rise to very similar clinical presentations, but fundamentally different mechanisms underpinning them. Therefore, arguing that the idea that we should all treat them the same is utter nonsense. And I think that is really, really important. And for the first time ever, she used the term viral associated wheeze, which we still kind of occasionally use in clinical practice. So really extraordinary, extraordinary work. Now, she also said, when I'm sitting in a clinic, and that is true for, when I'm sitting in a clinic, I observe kids who wheeze in early life and then stop wheezing, never to wheeze again. Those who wheeze throughout childhood, those who don't wheeze in early life, but wheeze later on. So she said, well, there may be these transient wheezers, late onset wheezers, and persistent wheezers. And a close colleague of mine, one of the sort of leading respiratory pediatricians in the world, whom I was very lucky to be able to persuade to come as a visiting professor to my unit in Manchester, and we spent many years sort of working together, tested this idea of different temporal patterns of wheezing in Tucson respiratory study in Arizona. But what he did was he said, well, I hypothesize that there are transient wheezers, and those would be the one who wheeze in first 3 years of life and not later on. That there are late onset wheezers who wheeze after age 3, and then there are persistent wheezers who wheeze throughout childhood. And lo and behold, he identified these patterns within his birth cohort study. We looked at this and we thought this is really a tremendously important contribution, but why would we say the 3 years or 6 years is important? And what is actually the pattern that is hidden within the data set? And to address this, as a first kind of collaboration in our stellar birth cohort consortium, when nobody believed that you could use data-driven techniques to understand patterns in medical data, we applied mathematical model latent class analysis to a large collection of data within one of our birth cohorts, ALSPAC, where we had data on presence or absence of wheezing over a period of first 6 years of life. And we applied model, and the model said, yes, indeed, there are different patterns, but they appear to be much more complex than sort of implied using hypothesis testing techniques. Indeed, there was persistent wheezing, transient wheezing, and late onset wheezing. But there were a number of these different intermediate phenotypes, starting, finishing earlier, later on. And this kind of data-driven, unbiased approach really kind of took off, and there were about 20 different studies who applied similar methodologies to data in different studies, all describing roughly similar phenotypes of similar name. The problem is that although these phenotypes share the names, they're very often fundamentally different in their nature. What do I mean? Well, the phenotypes were called the same, but their characteristics differed in, when the wheezing starts in late onset wheezing differed between age 3 to age 10. How did it stop? The prevalence was totally different. And we tried to address this to demonstrate that it will not, for those of you with background in mathematics, it will not come as a surprise that sample size, frequency, timing of data collection has major influence on number of types of wheeze phenotypes. And if you want to discover small phenotypes, you need large sample sizes. And even identical data points can result in different number of phenotypes depending on time intervals between time points. We also suggested what are the most informative time points, but have noticed that substantial number of children may be classified imprecisely. So we went then on to apply model now to the data from birth to adolescence across all our 5 cohorts. So this is now 16,000 individuals, which suggested that there are indeed 4 different patterns: persistent wheezing, late onset wheezing, transient early, transient late resolving. Now, when you talk about transient wheezing, people very often say, well, it's not really important because it goes away, it passes. What we have also shown very clearly is that in all of these different wheeze phenotypes, when we follow them long enough to their adolescence, that has major impact on their lung function, in that lung function is diminished in adolescence across all different wheeze phenotypes. Why is that important? Well, lung function in adolescence predicts not only respiratory outcomes, but also all health outcomes. Early deaths, cardiovascular deaths, metabolic disease, and all of the multimorbidities. So tremendously important. Not surprisingly, lung function was most impaired amongst those who wheezed persistently. Now the question is, are these different diseases? And the way we decided to approach this was by asking a simple question. Well, is genetic architecture of these different wheeze phenotypes fundamentally different? Because if genetic architecture of different phenotypes is different, they're likely to be different diseases. So we then went on to run, which is probably one of the first examples of multivariable or multinomial genome-wide association study in which we looked at genetic architecture at genome-wide level. So we had 7 million genetic variants, 16,000 kids, and then not looking at what are genes for asthma, but whether there's differential genetic association across these different Wheeze phenotypes. This is complex. Okay, so each of these circles represents all of the chromosomes, all of the genetic variants in different Wheeze phenotypes: persistent, transient, late resolving, and late onset. And what was staggering was that there was a set of independent SNPs associated with each one of these. So each one of these was associated with a group of completely independent SNPs. So we said, okay, I mean, quite extraordinary. Is there anything shared between these? And when we looked at that, so we compared persistent and transient wheezing. You know, blue is transient specific genetic signal. Red is persistent wheezing specific. Green is shared, but there's no green to be seen. Purple is shared, but in opposite direction, there's no purple to be seen. It is only when we relax this that we, you know, the p-value to something like 10 to -4 is when we see a little bit of shared genetic architecture, and that shared architecture maps to chromosome 70q21, which is a major gene for asthma. So not really surprising. We have also discovered 2 kits which were significant at genome-wide level, 70q21, confirming this asthma gene as actually being gene associated with persistent wheezing, but not others, and a brand new hit, never seen before on chromosome 9. So what on earth was this one? And what could we do thereafter? Well, we then honed in, focused on this, and did a number of other now basic science studies. So now we're moving from epidemiology, observation, genetic studies, complex mathematical modeling, into trying to understand mechanisms. 3 SNPs, 2 of which were located downstream of an exin-1 gene, and another gene almost reaching a genome-wide significance, but all of these 3 genetic variants were in liquid disequilibrium with each other, probably saying this is the same signal. We then did something that is called promoter capture high C to demonstrate that the lead SNP that we discovered actually physically interacted with promoter of an exin-1 gene. Wow, that is now interesting. And that's particularly true for CD4 T cells, which are relevant for asthma. So this is probably causative variant. We then did EQTL to demonstrate that this risk allele for wheezing is also associated with different expression of protein. Now it's becoming even more interesting. And when we looked at the biological function, well, an exin-1 interacts in genes which are enriched for asthma. So suddenly it makes sense. So what do we need now? Well, we need replication. But replication is impossible because our phenotypes, our clusters, can only be developed using these methodologies in large populations which would have exactly the same structure of data collection, which do not exist. So therefore, we needed to do further studies. We needed to turn to animal models and to careful laboratory studies. This is where another set of expertise, another set of human knowledge comes, and it is completely beyond my expertise. There comes my colleague, Professor Claire Lloyd, who then developed a mouse model of house dust mite induced allergic airway disease to demonstrate that the expression of this gene, an exin-1, is increased in lungs after we expose mouse to house dust mite, both in immunohistochemistry, but also when we look at mRNA. Then she did a killer experiments. She generated knockout mouse model without an exin-1 to demonstrate that if mouse does not have an exin-1, well, that mouse has a hell lot worse lung function upon allergen challenge than wild type mouse, has a lot more eosinophils, T cells in asthma, has a lot more Th2 lymphocytes, T cells in asthma, and all of asthma-related cytokines. So really, really exciting finding, demonstrating that perhaps annexin-1 has this pro-resolving feature which may regulate our immune responses to allergens, and it really now focuses us on using this as potential therapeutic target, or even target that will stop the progression of asthma through life course. Now, of course, all of this looks really cool and really great and really persuasive. We develop these phenotypes and say, here they are. The assumption is the clusters that we develop are internally homogeneous and stable. What do I mean with these terms? Well, internally homogeneous, that children within each one of these classes actually look very, very similar, if not the same, in relation to variable which we use to develop the model. Yeah, simple. Stable, well, whatever we do, however we perturb model, whether we use just those individuals with full data set or with imputed data set, same children always end up in same phenotype. Yeah, the basic you would remember, you would try to hope. Now, while this is a table, a sort of figure that we present in main paper, this is often buried in online supplement. And what this figure tells you is, what is the patterns of wheezing? So the color is when children actually reported wheezing from birth to age 18. And it's quite interesting, isn't it? Look how many children who are supposed to be never wheezers actually reported wheezing. Look how many children who are supposed to be transient wheezers actually wheezed between 10 and 18 years of life. And when you look at those two different phenotypes, late resolving and persistent, there doesn't appear to be a lot of difference apart from those with kind of late resolving appearing to have no wheezing at the latest follow-up. So they're not very internally homogeneous. How about stable? So we did a lot of modeling, reran the modeling, imputing a full data set to demonstrate that persistent wheezing and never wheezing are fairly stable. But all of these intermediate phenotypes, there's a lot of children moving from one phenotype to another when you use complete or imputed data set. And of course, if you want to do genetic studies in large samples, you need imputed data sets. Up to 25% of kids moved. So all of these models are based on the information of presence or absence of wheezing. And one of the postdocs in my study, who came from economics background and then did studies on the impact of poverty on health, said, well, you know what? Actually, it doesn't really, if you do poverty research, it doesn't make sense to say, well, this child was poor or not poor. It's not quite as simple as that. It is precise timing when you fall into poverty, the duration of poverty, number of spells of poverty. And she said, you know what, I'm going to try and develop a brand new model to model wheeze, not based on presence or absence, but much more holistic data on these complex spells of wheezing. And lo and behold, she did it very recently, developing these multidimensional variables of wheezing spells from birth to adolescence to include their duration, their sequencing, the extent of persistence and recurrence, to come up with architecture of wheezing that on the first hand looked similar. Never wheezers, late onset wheezers, transient wheezers, with two persisting classes, one being persistent and another intermittent. Now, how internally homogeneous are these clusters? Well, they are remarkably internally homogeneous. Not a single never wheezer has ever wheezed. All late onset wheezers start wheezing at the age of 10 and thereafter. All transient wheezers stop. The difference between persistent and intermittent wheezers is just really in whether you have wheezing all of the time or whether your wheezing is intersped by periods of health, which is so clinically intuitive. So they are very internally homogeneous. Then we said, well, how stable they are to data imputation, everything else. Well, they were remarkably stable. So running a model on full data set, and imputed data set resulted in only 0.3% of children switching phenotype, compared to 25% in latent class models. So are they different diseases? Well, they appear to be, because late onset wheezing and transient wheezing are not really related to genetic variants in 70q21. Persistent and two persistent categories are. And you may then ask the question, well, what the hell is childhood asthma? Well, this is where the problem lies, because childhood asthma is a mishmash of all of these different wheeze clusters. You know, a third of them are persistent wheezers, quarter are intermittent, quarter are persistent, and then 6% of children who never reported wheezing actually have doctor diagnosis of asthma. But their doctors see them every day, and I see them every year or 2 years of structured follow-ups. So they observe some patterns. So it is very interesting when you look at doctor diagnosis of asthma that even a third of wheezers who wheeze persistently do not have doctor diagnosis, which makes you wonder what actually asthma is. So where are we up to with this? Well, you know, wheezing phenotypes which we developed using Spell's approach are clearly much more intuitive than those which we derived using previous models, with great consistency and homogeneity, much more robust to data imputation. Will they be better reflection of underlying mechanisms? Well, you know, I hope to be able to tell you in a year or 2 or 3. This is the ongoing work, because we have to appreciate that 5% of kids with diagnosed asthma never wheeze, and a considerable proportion of those with persistent wheezing actually don't have a diagnosis. So we're now running much more complex model, trying to build in all of the symptoms and all of the information that we have to fully understand it. And that brings me to my last story, which is that of atopic march. And atopic march is not controversial. What is it? Well, atopic march is firmly in every textbook. This is how allergy develops during childhood. You start by having eczema in early life, progress to asthma, and then rhinitis. Now, the idea is, isn't it? It's a very sexy idea, because then you can identify children who will go on to develop asthma. They had eczema. You can try to intervene. I can tell you hundreds of millions of dollars were spent on studies trying to stop development of asthma in children with eczema. Also, it goes into guidelines. You should tell parents of children with eczema that their child may develop asthma. What I would say is, you know, my God, you know, they are more troubled with childhood eczema. They do not need to be freaked out by telling that the child may develop asthma. So where does the concept come from? Well, it comes from cross-sectional analysis at population level. A lot of eczema is present in early life. It's supposed to disappear. It's followed by asthma and then rhinitis. So we apply the standard methodology to our birth cohorts, and lo and behold, it looks almost the same. But the question is it really the same children with eczema in early life that go on to develop asthma? And what are individual patterns? And that brings you back to machine learning and the ability to do it at large scale in complex data sets. So again, we utilize the info.net and Bayesian inference in graphical models to look at the patterns of development of eczema, wheeze, and rhinitis in our birth cohorts over a long period of time, demonstrating very clearly that actually there are 7 different clusters of these diseases. 50% have none, and then there are various combinations. Actually, most children with wheeze only have wheezing, most children with eczema only have eczema, and they have other sort of different combination. Is there anything there that resembles atopic march? Well, not really. Eczema is a feature of 4 disease classes. Greatest number of kids with early life eczema never develop anything else. There is this interesting cluster of multimorbidity. So really, kind of, the idea of atopic march is just a fallacy, a myth that can be busted by learning from the data. Now, are these different clusters actually different diseases? We then again repeated it by looking at genetic architecture of these different clusters. Now, using polygenic risk scores. So we derived polygenic risk scores, including 130 genetic variants being associated with any disease in all of the previous GWAS studies, to demonstrate that there is really strong evidence that these different clusters have completely different genetic associates. This is fairly convincing p-value. And interestingly, one of the major genes associated with eczema, called filaggrin, although being associated with eczema classes, the strongest association was with this multimorbidity, with wheeze and with rhinitis. But, you know, it's very interesting, despite the clarity of the information, people love their simple models. Plenty of our colleagues that have then demonstrated that different children actually have very different patterns of development of allergic diseases. But the idea of March is refusing to die. So then we took a step back and said, okay, we will analyze this data in any way we can. Let's first ask the question, does co-occurrence between different diseases happen by chance? What do I mean? They're all common. So if there's 20% of eczema and 30% of wheeze at population level, by pure chance, you would have 7.5% having both. So we calculated trends in deviation between what we observe in population and what would be expected mathematically over time to demonstrate very clearly that single condition, albeit most prevalent at population level, are observed less frequently than by chance. What does it mean? There is a link between them. Two disease combination, quite frankly, it looks as if they occurred just by chance. But there was this multimorbidity of all of these different diseases, which was 6 times more often seen at population level than would be expected by chance. It's important. Then we looked at sequences of development, trying to see whether there's any pattern. And these are individual sequences in 4,000 children. And what you see is kind of beautiful abstract expressionist picture. There is no pattern. Actually, there are up to 350 person unique sequences across cohorts. So it's almost as if each child with atopic diseases have their own pathway. It's just kind of like Karenina complex. So there is no such thing as atopic march. We then applied further complex modeling to look at disease states, latent Markov modeling of transition of diseases across the whole of life course, which identified that there are really 4 disease states: low risk, children with mostly eczema, mostly wheeze, mostly rhinitis, and those with multimorbidity. Interestingly, children with mostly eczema also could have contemporaneous wheeze and rhinitis, but this is probably by pure chance. Model can pick it up. And you can also look at transition probabilities from one state to another, from birth to adolescence. And what is very clear is that this multimorbidity cluster is very stable. It forms by preschool age and then is stable to 18, and we've now modeled it to 35. It's very stable to age 35. And then you can look into probability of transition, and that's really interesting. So if you have eczema or wheeze in early life, you're most likely to remain having just eczema and wheeze, 60% probability. Transition from having disease to no disease is actually higher for wheezing, about 30%, than for eczema, where it is just 12%, so really sort of blowing away all of these ideas that most eczema disappears. Going from wheeze to eczema, almost non-existence. The most likely transition to multimorbidity is indeed from eczema, but the probability is only 20%, i.e., for an individual child with eczema, there's 80% probability that they will not develop subsequent other diseases. So what does it mean? Well, all methods, descriptive tests, frequentist methods, stochastic modeling, all have similar conclusion. Most children with early life eczema don't develop wheeze or rhinitis. Do not target children with eczema to prevent asthma. It's nonsense. Do not tell parents of children with eczema that their child may be at risk of asthma. It's nonsense. Of those who experience multimorbidity, almost nobody follows the sequence of March. There's vast heterogeneity, and different trajectories lead to multimorbidity. So yes, those with early life eczema at higher risk of having multimorbidity, but for that individual child that people see in the clinic, that risk is low. So the relationship about atopic diseases is not about marching It's a multimorbidity framework where no disease holds priority. Eczema is not important to any, more important than any of the other ones. So, in conclusion, I mean, as you can see, as I hope you can see, the idea that we can do silo, selfish science is absolute nonsense. No single data source or methodology can uncover complex mechanisms underpinning chronic diseases. Without understanding mechanisms, we will not improve patient outcomes. What we need is proper team science, cross-disciplinary, cutting-edge analytics based on large, complex, integrated data sets. We need birth cohorts, patient cohorts, randomized controlled trials, all underpinned by cutting-edge analytics, then progression to mechanistic studies in mouse models, in human tissues, again underpinned by cutting-edge analytics. And only through this effort, which is community effort, will we be able to solve the complexity of the diseases that are really impacting on human health across the globe. Only this approach will allow us to reform the taxonomy and approach in wheezing asthma from what we currently have, which is symptom-based to mechanism-based. To do this, we have to remove these ridiculous divisions which separate clinical research from basic science and data science. There are walls, there are silos. We say we're doing team science, but a lot of medicine is really firmly based in selfish science. Who is the first author? Who is the last author? I mean, who cares? We really have to abolish also the dichotomy of, oh, you know, hypothesis generation is better than hypothesis-driven approaches. No, they're both extremely valuable. We have to move towards more integrated approach where cross-disciplinary collaboration ensures proper scientific scrutiny and interpretation. And only fundamental shift towards team science will provide this catalyst to improve outcomes. And I have to say, Eddie, what you have done with BH Future Foundation is precisely that. It is generating a forum within which people with different skills, different interests can interact within the framework of potential team science to contribute to advancement of knowledge. And with that, you know, just think about what would Large Hadron Collider be without the community of scientists that are brought together around technological advances. You know, they say, oh, technology is going to save the world. Technology per se will not solve any of our problems. LHC, which is the most complex machine which we ever created, would just be, you know, an underground tunnel without the network of scientists who work around and who are dedicated to pursuit of knowledge. I'm really passionate about moving forward with the idea of team science and different types of expertise to feed in our understanding. And with that, there are plenty of people I have to thank. My friends and colleagues from Unicore Stellar Birth Cohort Consortium, Angela Simpson and Claire Murray, Raquel, George and Kate, Steve Turner, Sejal Saglani, Paul Cullinan, John, Graham and Hassan from Southampton. Fantastic data science team, Sarah Fontanella, Sadia Haider, Jada Oxel, Raquel Granell, friends and colleagues from Microsoft Research Lab in Cambridge, Chris Bishop and John Wynn, our friends and colleagues who do mechanistic studies in mouse models and in human tissues, Seb Johnston and Claire Lloyd, and all of my friends and colleagues from around the world, from South Africa, Ghana, Croatia, Turkey, across Europe, US, who have contributed to this. And with that, I thank you all very, very, very much for your attention.
Thank you, Professor Čustović. I think I can say for all of us a big round of applause, virtual applause. And we really enjoyed it. And thank you so much for this conclusion on how should science research work. I think it was really insightful. And also thank you for a phenomenal energy. We have one really, really good question. Selma, could you please unmute yourself and ask Professor the question yourself? Thank you.
Thank you so much. Thank you so much, Professor. It was a really amazing and interesting presentation. And I also work in the respiratory field. So I'm mostly working on lung cancer, but I'm also interested in connection between lung cancer and other lung diseases. And I'm mostly working on fibrosis and COPD. And I would like to know, would it be possible to use AI or machine learning for prediction of development of lung cancer with the patients that already have some sort of other lung disease?
Yeah, I mean, as you know, as being in that area, actually early detection of lung cancer is tremendously important.
Exactly.
And now you have, so it's a fantastic question. I really love it because, you talk about fibrosis and you talk about COPD. Actually, COPD, if you think asthma is in a mess in terms of understanding mechanisms, I mean, COPD is a much bigger mess. We call COPD, it really is based on impairment in lung function. So you have FEV1, FVC ratio less than 70%. I mean, that's kind of complete nonsense. I mean, isn't it? Complete nonsense. There are so many mechanisms. And you also know that, the original description of COPD, COPD is a self-inflicted injury. It's all of these people who smoke and, and then there is that worst thing of all. And then there's this inference, well, they got what they deserve, which I absolutely deplore. I absolutely deplore. Actually, all of our data now suggests very clearly that 3/4 of COPD start in early life and can be tracked back into early patterns of lung function. Whether, I mean, I don't know, probably you have seen this staggering data just published that what influences the risk of lung cancer in an individual is, okay, it is when they smoked, but it's whether their mothers smoked, which is really interesting, and whether their grandmothers smoked when they were pregnant with mother. We have this constant, it's cycle, transgenerational cycle of deprivation. And the problem that we have with COPD and lung cancer, they were always considered as diseases of the poor, and they received a hell lot of attention than others. What I would say is, yes, these methodologies are absolutely applicable, because trying to understand which subtype of COPD you have will then clearly have very, very, very differential risk in terms of likelihood of developing lung cancer. And then you can focus your prevention strategies on at-risk groups. People are trying to do that, obviously, already. How best to do it is really interesting. But the fact is, if your grandmother smoked and your mother smoked and you start smoking before age 15, the likelihood of developing lung cancer is 17.5 times higher. It is not 20% higher. It's 17 times higher. It's staggering. And these people are caught in the cycle of deprivation. Okay. And people say nothing can be done. I strongly disagree. Political action today is absolutely essential to break this. If you ask, it's almost like, we have terrible problem of air pollution in Bosnia and nothing can be done. Sorry, political action is needed today to break this terrible cycle and has awful impact on human health. Actually, sorting it out would be much easier than dealing with consequences, health consequences. But of course, nobody asks about that. They have better things to think about than health of population.
Yes, thank you so much. I also agree. I mean, all of these diseases are very complex, and what I'm looking at is tumor microenvironment. So a lot of different cells interact, and it's very, very individual. So you cannot have one therapy just for everybody. And I find this quite interesting. But I also would like to know, when you were talking about genetic stuff, I'm not so much into it, but you mentioned ANXA1 gene. I know it's also important for the initiation of cancer and also for progression for breast cancer. Do you think that we will see this also in patients with both diseases, or?
I mean, to be quite honest, it's, there's, 20 years ago, 25 years ago, I had a simple view of this. I thought, okay, so each of these different disease subtypes is unique and have completely unique mechanisms and genetic predisposition. I think that's gross oversimplification of truth. I mean, what you have is a limited number of mechanisms which contribute to developmental pathway, but pretty much on a sliding scale. So, it is the relative contribution of different mechanisms individual that then predetermines this developmental pathway, with clear understanding that this relative contribution changes over time. So yes, it's complex, and people say, oh, it's complex. Well, big deal. Let's embrace the complexity because we can understand it. And that's where I think, when people sort of raise issues about AI, well, sorry, it's whenever you get to machine learning or AI models, they need to be interpretable. So it's that interpretability that is critically important. So yes, I totally agree with you. And it's this understanding. So, with asthma and COPD, because they look similar. But so one is umbrella diagnosis and another is umbrella diagnosis. And they invented the third one, which is this ACOS, or asthma and COPD overlap syndrome. And you think, come on, guys, let's try and focus on mechanisms, translate it into biomarkers, into individual treatments for those with COPD, who are the ones who are going to develop cancer, who are the ones who are develop cardiovascular comorbidity, who are the ones who are going to develop metabolic comorbidity. Fantastic questions, Selma. I wish you all the best in your studies.
Thank you so much. Thank you so much.
Thank you both for this great discussion. I'm sorry you don't have much time. Selma also asked Professor if she could get some of your contacts to further discuss with you. Is it okay?
But of course, just, put my name in Google. It will take you to my email. Whoever needs any information, help, whatever, please do get in touch. Please do get in touch.
Thank you so much. Thank you. Thank you, Selma, also. I also have to read two more comments because they're amazing. Eddie wrote brilliant work. And also Ida Fazlic wrote, thank you for a wonderful presentation, Professor Čustović. I recently read a quote saying, competition happens at the bottom, people at the top are collaborating. And for me, this beautifully translates to the BH Futures Foundation initiative with the 3-minute thesis. We are all collaborating with our different scientific expertise for a better BH and global future. That's like greatly said. Thank you so much, Ida, for this.
Absolutely fantastically said. And I had this conversation with former Dean of Imperial who said, the scientific environment is Darwinian. And I told him, well, I'm sorry, but it's not competition that made us human, it's collaboration. So beautifully said, thank you.
Thank you so much, everyone. Thank you so much, Professor. It was great presentation, and thank you so much for answering all of our questions.